nucleic acids. Okay, what are nucleic acids? What is the function of nucleic acid? Okay, so coming to the topic, nucleic acid. You discussed about, you studied about the DNA and RNA, right? What is the function of DNA? What is the function of RNA? Or how they are made up of? All of them you studied in the lower classes, right? Yes. So we are moving to nucleic acids. Okay. So listen, nucleic acids are of two types we know. They are RNA and DNA, right? See, the transmission of inherent characters from heredity are due to the nucleus of a living cell. Chromosomes and nucleic acids present in the nucleus help in the transmission of heredity factors. What do you mean by that? See, generation to generation, the genetic information should be carried out, right? So how these genetic informations are carrying through the generations? They are done by DNA and RNA, means the nucleic acids, right? So what do you mean by DNA? DNA means deoxyribonucleic acid and RNA is ribonucleic acid. Okay, so we can say DNA is the genetic material, right? They are carrying the genetic informations. They are carrying the informations from genetic information from one generation or the parent body to the daughter body. Got it? So nucleic acids have that much importance in human body. Not only in human body, in every living organism, the nucleic acid have importance. Okay, then see, listen. What is the chemical? So what is the function of DNA? How they are working in our body? Everything we know because you studying in the lower class in biology, you studied about the function of DNA, RNA, everything, right? Now here we are going to study about the chemistry behind these nucleic acids, chemistry behind the working of DNA and RNA. What is the chemical composition behind the nucleic acid? Okay, so what is the difference between these two? See, listen. Pentose sugar, phosphoric acid and nitrogen contains heterocyclic compounds called bases upon complete hydrolysis of DNA or RNA. See, when you are doing hydrolysis of a DNA molecule or when an RNA molecule, what are the components you are getting? You are getting a pentose sugar. What do you mean by pentose sugar? Pentose sugar means, see, we discussed about the different category of carbohydrates, right? Keto sugar is there, aldose sugar is there, pentose, hexose. Okay, so pentose means it is a 5-membered structure. Okay, then pentose sugar is there, phosphoric acid is there, and nitrogen containing heterocyclic compounds. What do you mean by heterocyclic compound? Heterocyclic compound means normally we know what is benzene, right? This is benzene. This is the structure of benzene. Heterocyclic compound means, see, in benzene, in all the corners you can see carbon atoms, right? See, when one of the carbon atom is replaced by hetero atom like nitrogen, sulfur or oxygen, they are called heterocyclic compounds. Got it? Heterocyclic compounds means, it is the name itself, it is clear, it is cyclic compound. Okay, cyclic compound means they have a ring structure. Then what is heterocyclic compounds? See, instead of the carbon atom, there will be at least one nitrogen, sulfur or oxygen, means these hetero atoms will be there. So in DNA or in these genetic materials, you can see pentose sugar, phosphoric acid and nitrogen containing heterocyclic compound. It is clear. It is not sulfur or oxygen. Heterocyclic compound means they may contain sulfur, nitrogen or oxygen. But here in case of the nucleic acid, they are containing nitrogen containing heterocyclic compound. Okay, so they are normally called bases, nitrogen bases, commonly called nitrogen bases. Okay, then the sugar component present in DNA molecule is beta D2 deoxyribose, and in RNA the sugar is deoxy, means D-ribose. Okay, see, whenever you do hydrolysis for RNA or DNA, the components you are getting is always same. It is a pentose sugar will be there, phosphoric acid will be there, then a nitrogen base will be there. Okay, then what is the difference between this DNA and RNA? See, there is a structural difference is there. So we will come to that point later. See, before that, what is the important difference means? In case of DNA, the sugar component is there. Pentose sugar is there, right? The pentose sugar present in DNA is deoxyribose sugar, this one. In case of RNA, the sugar moiety means the sugar component present is this ribose sugar. Okay, so DNA, deoxyribose, RNA, ribose. Okay, then, so the components I told you, pentose sugar should be there, pentose sugar, then phosphoric acid, then nitrogen base. Yes, these are the three components of nucleic acid. So pentose sugar means they may be ribose sugar or deoxyribose sugar. If it is ribose sugar, it will be RNA. Means in RNA, the sugar present is ribose sugar. In DNA, the sugar present is deoxyribose sugar. Then the phosphoric acid is common in both. Okay. Then what about the nitrogen bases? So nitrogen bases means they are heterocyclic compounds. I told you. Heterocyclic compounds means the ring cyclic compounds containing nitrogen atom. Here it is nitrogen atom only. Heterocyclic means any atom, sulfur, oxygen or nitrogen may be there, but here it is nitrogen. Okay, then what are these nitrogen bases? Nitrogen bases are of mainly different types. See, adenine, guanine, cytosine and thymine. One more is there, that is uracil. Okay, these are the different types of nitrogen bases. What are the nitrogen bases? Adenine, guanine, thymine, cytosine, uracil. Okay, so all these nitrogen bases are not present in both the nucleic acids. Suppose DNA. DNA contains only adenine, guanine, cytosine and thymine. Okay, uracil is not present in DNA. Okay, so uracil will be present in RNA. Got it? Yes. So pentose sugar is there. It may be ribose or deoxyribose. So in DNA, it is deoxyribose. In RNA, it is ribose sugar. Phosphoric acid is common in both. And the nitrogen bases means we have different types of nitrogen bases. Adenine, guanine, thymine, cytosine, uracil. Then in DNA, the bases present are adenine, guanine, thymine, cytosine. Okay, is it clear? Then the structure is also important. See, this is the structure of ribose and deoxyribose sugar. So what is the difference between these two structures? See, in the first carbon, OH group hydrogen, same, right? In the second carbon, it is hydrogen. In ribose sugar, you can see hydrogen and hydroxyl group, OH group and hydrogen. But in case of deoxyribose sugar, both of them are hydrogen atoms. Okay, then if you are moving to the third carbon, here it is hydrogen and hydroxyl, same for both. And here you see CH2OH group is there, hydrogen is there. What is the difference between this ribose and deoxyribose? In the second carbon, the ribose sugar contains hydrogen and hydroxyl group. In deoxyribose sugar, they contain both hydrogen atoms. That is the structural difference. So these structures are also very, very important. In board exam, they are asking like, draw the structure of ribose sugar, draw the structure of deoxyribose sugar. They will ask like that. Okay, so we have to know what is ribose sugar, what is deoxyribose sugar. Okay, so the structure is important and this difference is also important. Okay, now we are moving to the detailed structure of nucleic acid. Okay, listen. See, nucleoside is found by, see, nucleoside. We are moving to different terms, that is nucleotide and nucleoside. What is the difference between nucleoside and nucleotide? Okay, see, in figure A, you can see it is nucleoside, right? So what do you mean by a nucleoside? So in DNA, as I told you, the DNA contains different components like pentose sugar, phosphoric acid, then One more thing is there, that is nitrogen base, right? Yes. Now, see, nucleoside is found by attachment of the base to the first position of sugar. See, here, this is the first one, nucleoside. Okay, so in this carbon, this is the first carbon, right? So in the first carbon, you can see the nitrogen base is attached. Okay, the sugar carbon in nucleosides is numbered as 1, 2, and 3 to make a clear distinction from bases. See here, because this is first carbon, second carbon, third carbon, fourth carbon. Okay, so in the first carbon, you can see the base is connected. So the pentose sugar and this base, they are together, they are combined together. That is called a nucleoside. Nucleoside. Okay, the pentose sugar and the nitrogen base, they combine. I will write here. Okay, pentose sugar plus nitrogen base. That is called a nucleoside. Nucleoside. Okay, got it, right? Then what is nucleotide? See, one more component is there. Pentose sugar, nitrogen base, and phosphoric acid is also there, right? Yes. So nucleotide means when at the fifth position of the sugar component, nucleoside is linked to a phosphoric acid. Okay, the joining of the nucleotides takes place by phosphodiester linkage between fifth and third carbon atom. Important, very, very important. See, nucleoside means the sugar and the nitrogen base. Yes, the first carbon, there will be the sugar, whatever the sugar, adenine, guanine, thymine, or cytosine, whatever it may be, it will connect at the first carbon. Okay, nucleotide means, see, the sugar is there, the basic component is there, and the fifth carbon contains the one more thing is there. I told you that is phosphoric acid. Okay, so that phosphoric acid is connecting at the fifth carbon atom. So all these three components together called a nucleotide. Means nucleoside plus phosphoric acid is called a nucleotide. Nucleoside plus phosphoric acid is called a nucleotide. Okay, so the structures are clear, right? So in the first carbon, the sugar structure, the first carbon contains the base and the fifth carbon is containing phosphoric acid. See, this is the phosphoric acid unit. Got it? The primary structure is the sequence of nucleotides in the chain of nucleic acid. Primary structure is the sequence of nucleotides in the chain of nucleic acid. Means in the nucleic acid, you can see what is the structure of DNA. You know the structure of DNA, right? Double helix structure. So in the structure, primary structure, the sequence of the nucleotides is the chain of the nucleic acid. Okay, in the double-stranded helix structure of DNA, what is that? See, double-stranded helix structure of DNA, the two nucleic acid chains are held together by hydrogen bond between pairs of bases. Again important. See, the DNA molecule contains two chains, right? Like this. They contain two chains. So this is called a double-stranded helix structure. Double-stranded means two strands are there. First strand, second strand. They're helical manner. They will join in helix structure, helical manner. Okay. So there is a double-stranded helix structure for DNA. Okay. So each strands are separate. It's not continuous like, see, it is not like this. Okay. They are separate strands. See, separate, not a single one. Okay, so each strands are made up of the same components that what we discussed in the previous topic. Okay, then how these two strands are connecting together? These two strands are connecting together by hydrogen bond. Hydrogen bond. Got it? Once again, in the double helix structure of DNA. This is the double helix structure of DNA. See, it will be like this. Okay. Yes. So here two strands are there. So the two strands are connected by hydrogen bonding. This one. Okay, so each strand is made up of this nucleotide, means phosphoric acid is there, nitrogen base is there, and the sugar means pentose sugar is there. So each strand contains pentose sugar, nitrogen base, and phosphoric acid. So how they are connecting through the hydrogen bond? Actually, through this nitrogen base, they are connecting through nitrogen base. They are connecting each other. Using this nitrogen bases, they are forming the hydrogen bond. Okay, but the thing is, see, what are the nitrogen bases present in DNA? I told you that is adenine, thymine, guanine, cytosine. So the thing is, they cannot see. Suppose this strand contains, here adenine is there. So when they form hydrogen bond with the next strand, they should be, see, the adenine can always combine with the thymine. Got it? Means if adenine is here, the thymine should be there. Means adenine and thymine can only combine together using hydrogen bond. Then what about this one? See, so adenine is combining with the thymine. Thymine is combining with the adenine. Then what about guanine and cytosine? See, if it is guanine, it will be cytosine only. So adenine base means adenine nitrogen base of one strand is forming hydrogen bond with the thymine base of the next strand. Okay, guanine in the first strand is combining with the cytosine of the next strand by hydrogen bond. Okay, so here suppose it is adenine, it will be thymine. Okay, that is the combination of that's how they are forming the hydrogen bonds between the strands. Okay, once again, nucleosides, see the DNA, right, means the nucleic acids means, first of all, we are going to discuss about DNA. DNA means they see, whenever you take a nucleic acid, there will be a pentose sugar, nitrogen base, and phosphoric acid. So we can convert the means we can split into two components, nucleoside and nucleotide. Nucleoside means this, see, the nitrogen base at the first carbon and the pentose sugar. Okay, nucleotide means this complete nucleoside is connecting to the phosphoric acid, means at the fifth carbon they will come. Who? This phosphoric acid. Okay, then in case of DNA, as I told you, there is a double helical structure. In the helical structure, the two strands are connected by hydrogen bond. While connecting with the other strand, see, the nitrogen bases are connecting the two strands. But the thing is, adenine can only combine with the thymine, guanine with the cytosine. Adenine cannot combine with the cytosine. Got it? So this is the combination of this nitrogen base. Got it? Okay. Now, what is the biological functions of nucleic acid? Next, we are moving to the biological functions of nucleic acid. Okay. So, what is the function of these DNA and RNA in our body? Okay. See, DNA is the reserve of genetic information and is the basis of chemical heredity. We know that because DNA is carrying genetic information, right? DNA is mainly responsible for the transmission of similar traits and identities among different species of organism. Means different characters. Okay, nature of different means different body parts like hair. Okay, so the DNA is the reason means they are carrying the information from one generation to another, or from the parent body to the daughter body. Okay, so DNA is carrying for, see, whatever the character, see, in our body itself, we have different character. Each of the characters are, means there is a DNA responsible for each of the characters in our body. Got it? So these informations are carrying out from one generation to other generation through this DNA, nucleic acid. Okay. During cell division, a DNA molecule undergoes self-duplication and the similar DNA strands are given to daughter cells. during cell division, everything you may discussed in biology classes, right? So during cell division, a DNA molecule undergoes self duplication and the similar DNA strands are given to daughter cells. Okay, that's how the informations are carrying from generation to next generation.